All right, it is now 12:01. Welcome and thank you for joining us today. I'm Eric Frigolis, Senior Associate Director of Major Gifts at the Feinberg School of Medicine. It's my honor to introduce our speaker, Dr. Peter Penzies, for today's presentation, Autism Across the Spectrum, From Understanding the Brain to Developing New Treatments. Dr. Penzies serves as the director of the Center for Autism and Neurodevelopment and holds the Ruth and Evelyn Dunbar Professorship in Psychiatry and Behavioral Sciences at Feinberg. He also holds professorships in neuroscience and pharmacology. Dr. Penzies is a native of Romania, where he completed his bachelor's degree in biochemistry at the University of Bucharest. After obtaining a PhD in biochemistry from the State University of New York at Buffalo, he carried out his postdoctoral studies in neuroscience at Johns Hopkins before joining Feinberg in 2003. Dr. Penzies' research is focused on the molecular and cellular mechanisms that control the development, remodeling, and disease of synapses, the connections between the brain cells. Results of this work help us understand the neurobiological substrates of brain disorders like autism, intellectual disability, schizophrenia, bipolar disorder, and Alzheimer's disease. He has been published in leading scientific journals with this research as well. At the end of Dr. Penzies' presentation, we have a lot of time for him to answer questions. Thank you to those who pre-submitted questions when registering for the webinar. You may also ask questions using the Q&A button at the bottom of the screen, and we will aim to get to as many as possible during the Q&A session. Thank you again, Dr. Penzies, for sharing your time and expertise. Please take it away.
Well, thank you very much, Eric. Let me share my screen first. Second. Well, hi, everybody. Good afternoon, and thank you for your interest in this topic. Autism, as you know, has been in the news a lot. It's been on social media. It's been, all over. It's been in politics. And so there's a lot of attention directed toward autism. The reality is that autism affects millions of people and affects millions of families. And there's a lot of interest, there's a lot of need for answers and solutions to many of the challenges that are raised by autism. And also there's a lot of need for clarity because of a lot of misinformation and misinterpretation of what has been happening in the media and the social media. So in my talk today, I would like to spend some time trying to clarify some of these concepts and trying to answer some of the questions that families may have. And thank you very much for submitting early some questions. I tried to incorporate as many of them in my talk as possible, but I'm sure there are other ones. So toward the end of my talk, I also wanted to introduce our work here at the Northwestern Center for Autism and Neurodevelopment, where we try to understand the basic mechanism of autism and try to develop new therapeutic avenues for autism based on this knowledge. A couple of points I would like to make before I start. So first is that in today's talk, I'm going to focus mostly on basic research on how we lay the fundamental understanding of the brain function and differences in autism using neurobiology, using scientific neurobiological models. I'm not going to talk so much about treatment options or treatment resources, but I could in the future, if you're interested, refer you to such resources. The second point that I would like to make is that this is a very dynamic area. So many of the things that I'm presenting today are still being debated, even the terminology, some of the specific words to use are still under debate today. And this is a very hot area of research. Things may change from today to next year. It's, whatever I'm going to tell you, it's not really set in stone. It's a dynamic field that's changing very rapidly. So with that in mind, the big question that many of you ask probably is like, what exactly is autism? So autism spectrum disorder is being defined as a group of neurodevelopmental conditions which are characterized by difficulties in social interactions and communication, along with restricted, repetitive patterns of behavior, focused interests, restricted and repetitive activities. Language impairment also used to be part of the definition, but has been removed from there. So one point here to point out is that it's a group of conditions. So it's not a single disease. It's a collection of conditions that are brought together under this umbrella of difficulties in social communication and social interactions, and also restricted and repetitive behaviors. It was first diagnosed in the 40s by Leo Kanner and Hans Asperger, and it was defined as autistic behavior. It was a very rare condition at that time. Kanner observed only a few patients, and those were the more severe types of autism. And Asperger described people on the higher functioning end with, moderate impairments, in particular in social interactions and repetitive behavior. So a very important aspect of this is that autism is a spectrum, and because it represents a very broad continuum, in almost like each individual has their own individualized form of autism. But broadly, the spectrum spans a continuum of abilities and challenges. On one end, you have people who only have mild disabilities, mild impairments in social and communication abilities, but are largely able to live independent lives, are highly verbal, have many skills. But on the other hand, you have people with very severe intellectual and communication difficulties. So I'm going to come back to this, and I'm going to discuss the spectrum in the context of some of the current debates, and also various needs and the genetic and biological basis of these different aspects of the spectrum. So another important thing to consider is that autism often doesn't come by itself. It's a feature of this complex condition that also includes as comorbidity or co-occurring conditions, intellectual disability very often. Maybe in about 60, 70, 80% of the cases, people who have autism also have intellectual disability. Sensory processing differences are actually very prevalent. ADHD, anxiety, OCD, sleep disorders are very common. Epilepsy, about 20, 30% of autistic individuals have epilepsy, depression, GI problems, and that's represented in the graph here. So often these comorbidities are actually posing a bigger challenge than the disease, the condition, the autism condition itself. And that is actually very important, as I will show you in the next few slides. So what are the challenges, strengths, and needs across the spectrum? This has been, again, debated very much these days because, as I mentioned, there are very different needs at the different ends of the spectrum. So autism is a very big umbrella term that covers these very different, groups of people with different needs and different challenges and different skills. So how do we go about this? How do we go about understanding this? And one way to do it is to really look at the specific needs each person has along the spectrum. So on one end of the spectrum, there are high functioning people also called level 1 autism, although, again, this terminology is debated quite a bit. And often these individuals have very little or no intellectual disability, but nevertheless, they have difficulties with social interaction, social cues, abstract language, behavioral flexibility and cognitive flexibility, and they also suffer from sensory overload. They also have often anxiety, depression, ADHD, meltdown, and unfortunately, often low rates of employment and difficulties in social integration, but also have strengths. So as I will point out in the future, and as you know probably from the literature and media, like Temple Grandin or Elon Musk, they have some autistic individuals in this range have unusual attention to detail, very strong skills in systematizing, visual thinking, just things just flash into their mind all at once, very strong memory, verbal and academic skills. And they represent about 30–40% of the autistic population. The best support for these people is integration really into workplace, sensory and learning accommodations, and neurodiversity informed approaches. In the middle of the spectrum are people with moderate needs. They have limited verbal skills, borderline or low IQ, co-occurring conditions, often like ADHD and anxiety, serious language and adaptive deficits may present more severe symptoms. Yes, these represent about 30–40% of the autism population. And these individuals require much more support. So they need functional enhancement, medication to control these comorbidities, therapies to improve social skills and communication skills. So these are in a more difficult, this group needs much more support. And on the other hand is level 3, which is also could be defined as profound autism. These kids or adults are nonverbal, they have severe intellectual disability, very often have comorbidities, epilepsy, depression, GI issues, and others. They are in very high need. So, you know, for these individuals, really a cure or a mode of prevention would actually be the most beneficial. So it's very important to point out these important differences in need, because in the debates about neurodiversity, this has come up very much. I'm going to come back to this again, but right now I would like to move on to some of the causes of autism. Yes. Yes. So there's also been a lot of discussion in the media about the causes of autism, whether autism is caused by various environmental factors, or there's an epidemic, or what is really happening. And the current science shows that autism is about 80% or more genetic, which means that genes account for this percentage of inheritability. But what is very important is interesting that it's not one gene of autism, but it's over 1,000 genes that play a role. And they interact in very complex and interesting ways. So this is called the genetic architecture of autism. Like, how was this genetic substrate underpinning of autism made up? And it's made up of many so-called rare variants in which you have one specific mutation in a person. But that mutation has a very strong effect. And that mutation in itself is maybe enough to cause autism in that person. But such cases are very rare. So maybe they're detected in 10, 20, 30, 100 people. And these are called rare and highly penetrant in mutations with large effect size. There are another type of so-called genetic variation, which is another more modern word for mutation, which are common variants. So these are small genetic changes that are present in you or I or everybody, and they have to add up. Each of them have a very small contribution, about 1% increase in risk or reduction in risk. So a lot of these have to add up to tip your risk over into the autistic risk. And this also relates to this population distribution. So people with more severe forms of autism are often characterized by these rare variants, rare mutations, and people with milder forms of autism have these common variants, which often combine actually with environmental factors, like in these gene and gene environment interactions to bring about autism. Another aspect here to clarify is that genetic doesn't necessarily mean inherited. So these genetics are mutations that occur in the genes, but some of them, actually many of them, are spontaneous and they occur in the germline. So they are not inherited from the parents. So the mutation can occur spontaneously, and this could have important consequences for example, genetic testing. If a child has autism, parents often ask, well, could I have more children? Do they inherit it from me? And in many cases, if the mutation is spontaneous, then it's okay to have more children, and it hasn't been inherited. But in other cases, these genetic variations could be inherited and could be masked in some ways in the parents. For example, they could be recessive or some other things. Another interesting finding from these genetic studies was that many of these same genes that participate in autism also participate in schizophrenia, bipolar disorder, epilepsy, ADHD, OCD. So what this shows is the biological substrates of these disorders, of these conditions, are common. So, as I will show you, these genes affect brain development, and that altered brain development could lead in many different directions. So, I think I deleted a slide, but okay. So now I'm going to move to environmental causes. There's also been a lot of debate recently in the media, and this issue has also been politicized about the role of environmental factors. So as I mentioned before, about 80% of risk is genetic, and up to 20% risk by difference is environmental. So the consensus tends to be that the environmental factors are not sufficient by themselves to cause autism. So they are not a sole cause. And on the other hand, environmental interventions like supplementations or various conditions are unlikely to be by themselves sufficient to be a sole cure. But in general, environmental factors interact with genetic vulnerability, genetic risk that originates from some of those genetic variations, and combine with this risk to tip the balance toward the expression of an autistic phenotype, so to say. So several environmental factors are quite well established, and they are shown here. So a lot of them are related with gestation. So, things that happen in the womb. One important factor is maternal infection or fever of the mother during pregnancy. Also, obesity in the mother, diabetes, exposure to medication like valproate, which is an anti-epileptic medication, stress, and other issues related to birth complications or preterm. So all of these factors could add up to a pretty big contribution, environmental contribution. Air pollution has been shown to play a role. One interesting factor is advanced paternal age. So as mutations in the germ cells of parents accumulate, as with older parents, there's a higher risk that such de novo or spontaneous mutations occur and are present, occur in the sperm, and then they are transmitted to the child. But one aspect of this is that many environmental factors have not been characterized. So we know that there's a kind of a missing environmental contribution, but it's very difficult to measure. How many different things are you exposed to during life? Like, how many different ingredients in your food or, clothes could contribute to autism? It's very difficult to measure. But some environmental factors are protective. So, for example, prenatal supplementation with folate has been actually shown to be protective, and that's very well validated. So the environment could act both ways to enhance genetic risk, but also to reduce genetic risk. I want to touch on some controversies that have been in the media recently. So Tylenol has been discussed as being, a cause of autism. So the current evidence shows there's very weak epidemiological evidence. There are some signals, some weak signals that are associated with Tylenol and increased risk for autism. But this is not a causal proof. The most likely relationship is actually maternal infection. So if there's the mother has a cold or flu or some viral disease, that in itself could increase risk for autism. And the mother takes Tylenol for the fever. That is an incidental, as a coincidence. Another discussion was about leucovorin, which is folinic acid. So this is more interesting potentially because folinic acid or leucovorin is related to folate, and prenatal folate, as I mentioned, has been shown to be protective. So now prenatal folate and leucovorin administered postnatally act in different ways, but nevertheless, they target the same pathway. And there are actually a few small clinical trials showing that postnatal folate or folinic acid actually could be beneficial in some autistic children with folate receptor autoantibodies and cerebral folate deficiency. So these are small clinical trials. There's a potential here, but at this point, we can't say yes or we can't say no. We just need to study it more. There is a slide. Sorry, there's a slide here that I missed. I'm going to come back to it. And that talks about epidemiology of autism. So I'm going to see if I can, just 1 second. I don't know exactly what happened to it. I'm going to log out for 1 second and share my other presentation. Okay. Sorry for that. So now we understand that the genes play a role and the environment plays a role in autism. And so how do they play together? How do they actually lead to autism? So what we know so far is that these rare genetic variants and the common genetic variants which interact with environmental factors, they represent many, many genes, like 1,000 genes. And it could be that the genes that play a role in autism in one person are very different from the genes that play a role in autism in a different person. But what we know now is that these genes converge on so-called genetic and molecular pathways. So these are networks of proteins or genes that interact with each other. And as they interact with each other, they control specific biological processes. And we find out that they converge on two large groups of biological processes: control of gene expression regulation in the developing brain, and control of formation of synapses or brain connections between brain cells. So what do these pathways do? Well, practically they control how the nervous system, the brain, develops. And they control early stages of brain development, how neurons or brain cells differentiate, how they migrate to their correct place in the brain, how synapses or connections between brain cells form, and also how these immune cells within the brain called microglia interact with brain cells and how they shape these brain circuits. So altogether, these processes contribute to the large-scale brain development. And if there are, and ultimately these lead to the wiring of the brain and formation of brain circuits, which underlie all of our behaviors and the thoughts. So if there are some small defects in any of these genes, then these pathways don't function properly. These processes don't happen properly. The brain doesn't get wired as in a neurotypical person. So we get these functioning circuit level differences, which result in behavioral and cognitive differences. So this leads to some more integrative neurobiological models of autism. And I'm not going to focus now to discuss psychological models or social models, but focus only on neurobiological models of autism. So one important model refers to autism being a type of synaptopathy or a synapse dysfunction related condition. And as I mentioned, many of these genes contribute to the formation and function of synapses. Another such model focuses on these interactions between microglia or neuroimmune cells and how these microglia shape the connections between brain cells. Yet another model focuses on early development, early brain development, and migration of brain cells in that stages. So all of these together are the basis of a fourth model, which refers to connectivity between brain cells within the same brain region, or short-term connections, which tend to be increased in number in autism, which is called hyperconnectivity, so a local network hyperconnectivity, or and reduced connections between long distance connections between different brain regions, and this would be hypoconnectivity.
Hey,
All
Peter, I'm
right.
sorry to interrupt, but your presentation is not up, so I just wanted to let you know.
Oh, it's not up?
Nope.
Oh, God, so I've been talking for so long. Oh, I'm sorry for all that.
Perfect. We see it now.
Okay, yes. So another theory focuses on the balance between excitation and inhibition, between excitatory and inhibitory signals in the brain. And this leads to differences in information processing, but also in seizure susceptibility. So what can we do with all this genetic information at this point? And there are a few things that we can use right away and how it helps us right away. So genetic testing is fairly common, is relatively accessible. It's not cheap, but it's accessible these days, and it could identify about 20% of the cases. So in about 20% of the cases, one could fairly precisely pinpoint to the specific genetic mutation. although currently it's not used as a diagnostic tool. But it can help families make decisions about having more children depending on the type of mutation. So if a mutation is a spontaneous de novo mutation, then it's okay to have more children. It can also reveal additional medical conditions that could come up later that the families can be prepared for. So, for example, seizures, obesity, or others. And it could inform treatment strategies. So some patients with some mutations tend to respond better to some specific medications versus others. And as more and more information about this builds up, this information, genetic information, could be very useful. And there are also these gene-based clinical trials that are emerging now with specific treatments geared toward one specific gene. So as opposed to targeting autism in general, these are clinical trials that target patients with one specific mutation. Also, families tend to group and form family advocacy group or foundations based on specific genes and carriers, individuals with specific genes. And for example, is the Gene SCN2A Foundation, or SYNGAP Foundation, TRIO Foundation, Phelan-McDermott Syndrome Foundation. And parents in these foundations can exchange information, can provide mutual support. Kids with mutations in the same gene tend to have some similarities and some individualized specific needs. So these type of foundations could be extremely useful for parents. They could also influence policy, could fund research into that specific disease, or could participate in specific clinical trials related to that specific rare condition. And together, they could partner with academic labs, like it's happening actually in our center, and contribute to the formation of biotech startups. So together, they could form a very powerful translational system. So this also leads to a new approach to thinking about understanding and treating autism. So traditionally, treatments have been guided by symptoms. So like target autism in general based on some broad criteria. But the new approach is called genetics first. So it looks at individuals with one specific genetic mutation and tries to develop specific targets, specific treatments, and design specific clinical trials for that group. And that makes development of these biologic pathway directed treatments for genetic subtypes. The clinical trials are faster, easier, smaller, more efficient. And that I see as being one of the potential new directions for research. So I am missing one slide, but I wanted to talk about this. And that's a very important question that comes up a lot. It's like, is there an autism epidemic? And what is happening? As I mentioned, some individuals with autism have very serious needs and have severe forms of autism. And others have more moderate or modest needs and could be quite functional in some ways. So it seems that the majority of new diagnosis comes from the side of more higher functioning individuals. And the number of the incidence of cases of lower functioning autism has stayed more or less constant. So what this suggests is that there's actually an increased recognition and diagnosis, including self-diagnosis and broadening of diagnostic criteria that includes more and more people, but more on the functional end of the range. So I think this is very important to note because it may sound like an epidemic, but in reality, it may be just more and more people that recognize that the conditions they have, the difficulties they have, as being autism as opposed to previously. The other aspect I would like to point out is the different strengths and challenges. So as I mentioned, some people on the autism spectrum have enhanced abilities. So, for example, enhanced memory, enhanced visual imagination, and enhanced attention to detail and systematizing. So this is also very important to consider when considering the strengths and the challenges across the spectrum. So now I would like to speak briefly about current treatment options and resources. And again, there's quite a bit of debate about whether we should treat or cure autism, or we should just provide support for individuals to be who they are. And I think that the agreement in this is that we should treat what causes the difficulty and the suffering. And the ultimate goal should be to improve independence and quality of life, not to erase identity. So we would like to help people to the extent that they need help. And some people with autism may need only accommodations and support, but others may need something closer to a cure. So currently, there is no cure for autism. What we can do now is to manage clinically these comorbidities. So, anxiety, sleep, ADHD, epilepsy, GI problems, and they cause quite a bit of difficulty for the individuals and also for the families. And looking specifically at autism, there are behavioral therapies and speech therapy that help patients improve their social and communication skills. And these could be quite successful, especially in a high functioning and a moderate level functioning individuals. But they're extremely time consuming. It's a full-time job to provide this type of treatment. It's extremely expensive. So that poses a problem of how accessible these treatments are for many people. And also there's a challenge here because there's been a lot of misinformation in hyped up treatments and a lot of unreliable sources in the news even, or social media. So I would like to point out briefly a few reliable sources of news. So the Simons Foundation is a private foundation that contributed extensively to autism research, and they have a publication called Spectrum, which I think comes out every week, and it can be accessed at this website. And it digests all the news about the new developments in autism, and it's a reliable site. Also, Autism Speaks is a prominent advocacy group. The National Institute of Health, which is a government agency, has updated information. And about clinical trials, is a clinicaltrials.gov website. So speaking of pharmacological treatments, of course, we're thinking about a pill to help children or adults with severe forms of autism. And there are currently about 460 such clinical trials for interventional new drugs. And this is a pie chart of the different stages they're in. And most are repurposing existing drugs. So, things are coming down in the pipeline. But I think that there is some promise here. And I would like to point out this one example, which is a recently FDA-approved drug approved for Rett syndrome. It's called Daybue or trofinetide. And it's the first medication that actually treats some of the core symptoms of a neurodevelopmental disorder. So not some of the comorbidities, but some of the core symptoms, such as intellectual disability and autistic features. So itself is not a very potent medication. It's not a magic solution, and it has some pretty difficult side effects. But it is a proof of principle that we can improve actually the core symptoms of a neurodevelopmental disorder, and even when this drug is given after birth. So we hope that in the future, hopefully in the close future, such drugs could be developed for various forms of autism as well. So now I'm changing to what we do here at the Northwestern Center. And we established the center almost 10 years ago as a center of research, the Center for Autism and Neurodevelopment at Northwestern. And our mission is to understand the biology of autism, to understand the genetic, neurobiological information of how brain develops and how it develops differently in individuals with autism, and to use that knowledge to develop new, safe, and effective treatments to help those in need. And we are a research center, so we're not a clinical service, we're not a clinical provider. Lurie Children's, on the other hand, and UIC or Rush have clinical services for autism. But we focus specifically on the science and in the idea of, creating future therapies. We bring together expertise from many fields because autism is a very difficult, complex issue that needs a broad range of expertise. And in this sense, it's been funded to unite Northwestern's strengths in neuroscience, psychiatry, genetics, bioengineering, and others. It includes about 30 faculty and working in all of these disciplines and departments, and over 200 students, postdocs, and technicians who work in these labs on projects related to autism and other neurodevelopmental disorders. This is our website, so I invite you to take a look and look at the different links there. So I just wanted to go over briefly on our strategic plan or research pipeline of how we want to go from individual genes to future drugs. And this is a cartoon of it. So we start with genetic mutations. We test, validate the biology of these genetic mutations in mouse models, or preclinical mouse models and cell models, and then use this information to develop experimental therapeutics, which again, we go back now and test in these models, and then hopefully we will put in clinical trials. And these are the steps of the process. So first, we identify genes of interest. And as I mentioned, we don't focus on autism as a broad category, but we focus on these sort of so-called monogenic or rare syndromic forms of autism that are associated with specific genes like TRIO, SYNGAP, SCN2A, ENC2. And then we move to study the function of these genes and why they are disrupted in autism in test tubes or in cultured brain cells. And then we make mice, laboratory mice with the mutations in these genes, the mutations that are also present in patients. And in those mice, we can study as a model system, as I'm going to mention, I'm going to show you in a few slides. We can also derive human neurons, and these we can take brain cells, or we can take blood cells or skin cells and from patients that carry the mutation and grow them into neurons and culture them in a dish. So this is a model of a disease in a dish. And then we can use this information to set up screens for drugs, for drug candidate molecules that we can use to normalize the function of these genes or these networks that they are involved in. And our hope is to set up, take these drug candidates into clinical trials. And then these clinical trials would be these small clinical trials for orphan diseases. And the FDA has incentives for targeting orphan diseases or small rare diseases. And it gives our drug candidate an orphan drug status, which makes clinical trials smaller. So you can do a clinical trial with 20 people as opposed to 200 people. So it's much cheaper, faster, more feasible. And the goal is, once we find some drugs that work in these rare disease conditions, expand the indication to a broader form of autism, depending on the relationship of the genetic pathway. So now, very briefly, I wanted to move to some of these examples of what we do. So we use several cutting-edge technologies to understand the biology of autism. So one of these is using mouse models, because once we identify the gene, we want to study what it does in the brain. So we can make genetic mutations in these mice, and mice have a lot of pups, so we can have a lot of mutated pups. And then we can study in these guys' behavior. So here, for example, we study social behavior. Here we have this cage with a mouse, and then here there's hidden a new mouse, or here there's an empty cage, an empty little box. This mouse prefers to hang out with a mouse because mice are social. But when we make a mutation in this mouse, it actually prefers the empty cage more than the other mouse. Or we can also study vocalizations in these mice. I don't know if you can hear me, but these mice make some ultrasonic sounds that we can study. And these sounds are altered by mutations. We can also use microscopy to peer into the mouse brain and look at the activity of many, many neurons, as you can see in this movie, how the neurons fire in the brain of a live mouse. So we can compare mice with a mutation in an autism risk gene with control mice and see what these differences are. Also, we can do optogenetics, where we can manipulate the behavior of these mice and make them perform or stop behaviors that are related to autism. Another very important technology is these stem cell-derived neurons or organoids. So in this, we can take skin or blood cells from patients. We can reprogram them, and these cells would have, the so-called stem cells would have the same mutations as the patients, which we can then differentiate into neurons and culture them in a dish. So these are brain cells coming from a skin cell from a patient, actually. And then these we can develop further to form these brain in a dish or organoids. And this is an actual organoid from one of our labs. And we can grow these in a dish. And mice and these organoids we can use to test and develop these drugs. So ultimately, the goal is to develop new medicines. And once we know the biology, we can use that biology to set up screens or use computer modeling to try to identify these small molecules that fit into those proteins and can be developed into drugs. And the goal is to develop precision medicines for subtype of autisms and then extend this to autism in general. So as closing thoughts, I wanted to say that autism is a spectrum of brain development related conditions with wide differences in abilities and challenges. The support and treatments should be tailored to the specific needs. It's no one size fits all. It is mostly genetic, but the environment also plays an important role. Comorbidities are also common and disabling, often more disabling than the autism itself. So now we have very powerful tools to understand the genetic and neurobiological basis and how this is beginning to transform how we think about diagnosis and treatment, like the genes first approach. And new personalized medical therapies and are urgently needed, and we are working on them here at Northwestern and in other places. So thank you very much. Thank you for your attention, and I'm happy to take questions.
Hi, everyone. I just wanted to say thank you, Dr. Pences, for your informative and thought-provoking presentation. It is 12:50, so we're likely going to extend the time of this webinar until 1:15 if people are able to stay on. If not, we will send a recording next week with the questions and answers. We received many thoughtful questions both in advance and during today's session. Thank you to everyone who submitted one. We'll try to get to as many as time allows. Dr. Penzies, do you have any thoughts on why autism spectrum disorder is being diagnosed more often now than 20 years ago? Is it more about awareness and a greater understanding of the condition, or is it attributable to environmental factors?
Yes, I mentioned in brief earlier, uh, there's quite a bit of uh, reason to believe that the increased diagnosis, increased incidence comes from broader diagnostic criteria and more awareness. And often people now, adults, mostly on the higher end of the spectrum, higher functioning end of the spectrum, self-diagnose themselves. Uh, and that contributes a lot to the increase, especially on the higher functioning end of the spectrum. On the other hand, on the lower functioning end of the spectrum, there hasn't been so much change in incidence. So that makes us think that overall, the number of autistic people or the incidence of autism is not changing overall, but more and more people are diagnosed because of the criteria, because the criteria are broader. Yes, that's my thought.
Thank you. Next question is, in your presentation, you mentioned comorbidities that overlap with other neurodevelopmental conditions. How do ADHD and autism overlap, and can it be difficult to identify the dual diagnosis?
Yeah, so there's quite a bit of underlap, and I had numbers, and I think it's something about 35 or 40% of kids with autism also have ADHD. So I would like to say about this that many of the risk genes that lead to ADHD are also risk genes that lead to autism. So there might be some overlapping neurobiology there. Uh, ADHD in itself doesn't entail difficulties with social communication, but ADHD often comes along with autism. So I think that the relationship goes more from autism being associated with ADHD more often than ADHD with autism.
Thank you. Next question is, what is the difference between autism and Asperger's? Is Asperger's a distinct condition, a part of the spectrum, or a specific mutation?
Yeah, so I'm gonna try to find my one slide that I missed, and I don't know exactly what happened to it because that slide had something related to this. And but the point is that oh yeah, okay. So I'm gonna share this now here, and I don't know why it disappeared. Can you see my slide now?
No. You can see it now.
Okay. So yeah, I don't know why this disappeared, but this is the population distribution of, let's say, one trait relevant for autism, social intuition. And what we have here is sort of a bell-shaped distribution, like a Gaussian bell-shaped distribution. And in blue, we see the population at large. So this one we would call neurotypical. And on this tail, these are the individuals with autism. So if we blow this up a little bit more, sorry for it being fuzzy, what we see is that on this end, we have these high-functioning individuals, and they overlap actually with the neurotypical population. So but they overlap with a sort of lower functioning end of the neurotypical population. And then there's another peak here. These individuals with moderate and severe forms of autism, which almost form sort of a distinct peak. So initially, when Leo Kanner diagnosed autism and named it autism, the individuals diagnosed were in this red group with severe forms. And when Asperger found his first in patients, they were in this green high functioning group. So this high functioning group overlaps quite a bit with a neurotypical population, but it's more toward the lower functioning end of it. So there's been a lot of debate about whether these are two separate groups or they are part of a continuum. And there are some differences in the different types of genes that participate in these two groups. There's clearly a difference in severity. There are differences in needs. So now they're all combined in one large, definition of umbrella definition of autism. But it's also possible that, another way of approaching it could be to separate the higher functioning, the older Asperger's definition from these lower functioning, more severe, monogenic, syndromic types of autism.
Thank you. In your presentation, you discussed the three levels of the spectrum. What is the distribution of individuals across these levels? I've heard this is equal probability, which seems implausible.
Yes, let me just find that slide here. Yes, so am I still sharing it?
No. We see it now.
Yes. So it's not very easy to estimate this. I think it's much more easier to estimate the level 3 more profound autism, which represents about 15 to 25% of cases. Level 2, again, represents about 30 to 40% of cases. And high functioning is about 30 to 40% of cases, but a large just fraction of the new diagnoses come from this high functioning autism group. So I expect in the future that the proportions will change because, as I mentioned, this low functioning level 3 autism stays fairly consistent, while more and more people are diagnosed or self-diagnosed in this high functioning group.
Thank you. Is there a specific kind of maternal infection that affects the risk of autism? Can a common cold in pregnancy be a risk factor for autism, or are only certain or more serious illnesses risk factors?
So flu has been associated, influenza has been associated, other types of like measles, other types of, actually not measles, other type of infections have been associated. In general, more severe, and the effects are different from individual to individual. Some people may be affected more than others.
Thank you. How do you obtain statistics about estimated group size or other demographics? Are these self-reported? Is information only collected from individuals who have been diagnosed with autism? And how do they account for potential underreporting, such as individuals who never obtain an autism diagnosis?
Yeah, that's the major challenge here, that the way diagnoses are made, and I think that's another slide that I may have missed. But yeah, diagnosis. No. Yeah, so diagnosis normally would be made by a specialist, and it's made based on the DSM-5 clinical manual, and it's based on social communication, repetitive behaviors. There are some standardized tests, so this whole testing process would take about a day or so and would cost a few $1,000s. Unfortunately, there is no objective measure like a blood test or a brain scan. Like, for example, for diabetes, we can, measure something. Here, there's a lot of subjectivity. And what makes this more complicated is that a lot of people don't have access to this, specialized, uh, you know, testing. And even if they do, sometimes wait lists could be like up to a year or maybe longer. So a lot of these, assessments are assessment by parents, assessments by primary care physicians. Even, for example, in the news, there was one study from South Korea where the investigators sent out this questionnaire to parents, and the parents had to fill it out depending on what they felt. But parents, there's a lot of subjectivity. So, it's very difficult to get, clear numbers. What I'm showing here is based on the CDC data, Center for Disease Controls and Prevention from the US. And it's, more or less, as accurate as it could be. But these numbers could differ very much based on, who reports and how they report. There's underreporting, under-recognition in girls, for example. So we may actually be missing many cases. Yes, these are some of the challenges.
Thank you. What is the best way to battle misinformation about autism? And how do we ensure that treatments don't eradicate the benefits of neurodiversity?
Yes, so I mentioned a couple of sources, and I think first is to sort of build a knowledge base for each person, each family, to really understand some of what's going on, some of the neurobiology and some of the epidemiology that I discussed here today as a reference point. And we know that, for example, and the environment plays a smaller role than genetics, and in general modulates or modifies genetic risk. And no one environmental factor is strong enough to have a major effect. So if you know that, and you understand enough about why is that way, what is the evidence supporting that? When you see in the news that, let's say Tylenol causes autism, then you're going to start questioning it. It's like, oh, it doesn't fit in my, in what I know already. And the second is to go to some of those sources that I mentioned, the Simons Foundation, Autism Speaks, NIH, and look at what they publish on this. And sort of get an idea of what we know, what the big picture is now. And when you see something new that really sticks out, that doesn't quite make sense, even some claims of something, one obscure factor causing or treating autism, then it's just going to seem odd to you, and you can then read about it and see beyond the hype and see beyond the misinformation. And I'm sorry, what was the second part of your question?
It was, how do we ensure that treatments don't eradicate the benefits of neurodiversity?
Yes, so I mentioned in one of the slides that we need to tailor treatments to the specific needs, and people on one end of the spectrum who have, let's say, are higher functioning but have difficulties with social interactions or being organized, but they have strong skills in other aspects. They need very specific treatments which would improve their social skills but wouldn't erase the autism. But people on the other end of the spectrum who have very profound autism and who need 24-hour support, they may need some serious interventions like gene therapy or medications. And in that case, it's very valid to talk about curing autism for those subjects. So, we have to tailor the interventions to the needs.
Thank you. What is the optimal age for intervention?
So the earlier, the better. That's very clear because the brain, develops when early on after birth, the brain develops fast, and then those changes are logged in. But because it grows so fast, it changes so fast, that's also a time when we could intervene. The challenge there is that, most kids are diagnosed between the age of 2 and 4. Uh, so we can't really intervene before. If some kids have let's say the parents undergo genetic testing while the child is in the womb, and the child has a mutation which is, predicted to lead to autism, the severe form of autism. There's, similar situations are, considered for other neurodevelopmental disorders like Rett syndrome, and that there are currently gene therapies that are being developed, still not available, but there could be the possibility to intervene prenatally and, correct the genetic defect. Or, our hope is that, we could develop a medication, a pill that one could take, maybe even prenatally. So again, there are some risks associated with that. But the point is to intervene as soon as possible. Some interesting things about animal model studies, and the example I gave with this drug, Daybue, is that the drug, for example, is being tested for Rett syndrome, and it seems to show benefits even in adults or teenagers with Rett syndrome. So what this shows is that even if the nervous system is already on a different course, it's been different than neurotypical, we can still use some interventions and normalize parts of that behavior and the function. So that gives us hope that there could be treatments that could function even later, maybe even in adults.
Thank you. Is there an understanding of the incidence being much higher in males than females?
Yes, it's been the numbers seem to be 4 times higher in males and in females. It's not very clear why. One contributor, and it's probably many factors that contribute there. So one contributor could be that the specific expressions of autism in girls is different than in boys, and that leads to under recognition in girls. That is part of the. But it could be, different mechanisms are acting. Could be girls could have more genetic resilience or biological resilience to autism. So that same genetic mutation may lead to autism in a boy, but the girl with the same mutation may not have autism, or maybe not be as severe. And we don't know whether it's sex hormones, whether it's some other biological processes, but that is investigated quite intensely, because if girls have some protective mechanisms and we can learn from that, then we can apply that to all subjects with autism.
Thank you. Can imaging or scans show any evidence of autism?
I'm sorry, what scan?
Can imaging or scans show any signs of autism?
So there are some small but significant differences between brain scans in patients, or especially with more severe forms of autism and neurotypical individuals. And when you group individuals based on these narrower groups, based on specific genetic mutations, these differences in brain scans or EEGs, for example, become even more pronounced. They are not at the level of being diagnostic. So it's at this point, we cannot use, for example, a brain scan to tell a person with autism from a person who doesn't have autism or maybe has something else. Uh, but for scientific reasons, brain scans and EEG could be a good way to study patients with autism or potentially biomarkers for treatments. If we have treatments, then we can watch the changes in these biological measures like brain scans or brain electrical activity levels as a measure of whether those drugs work in the clinical trial.
Thank you. Is an IQ test required to have the 3 levels assigned to a patient?
I don't really know about the specifics of how these tests are administered, and I think even this grouping on 3 levels is not fully accepted by everybody. But I think an IQ testing or IQ type of testing is part of the basic neuropsychological assessment, which would then be used by a clinician, to group patients. And it's also important to point out that patients at the more severe end of the spectrum or level 3 are always associated with intellectual disability or an IQ below 70, and also other comorbidities. So IQ testing is an important part of grouping patients.
Thank you. Have there been any developments in preventative measures against skill regression or supplementary measures to support autistic individuals suffering from skill regression upon diagnosis?
So it's just intense, the same behavioral therapies that are applied, for improvement of skills are used for skill regression as well. I'm not aware of any additional treatments, but I may be just not well informed about them.
Thank you. Are there any recommendations that potential parents be tested for genetic markers in advance to be aware of the possibility of autism in their offspring, with the ability to plan ahead in terms of resources, gathering information, awareness, etc.?
So that's an important question, and it's not very easy to answer. So I'm just going to go to this other picture again, other slide about the genetic variation. So as there are 4 types of these mutations here. You have these rare mutations and then common variation, and then these so-called de novo spontaneous mutations and inherited mutations. If you perform genetic testing in parents, you might catch these rare variants that did not lead, rare variants that are inherited, that did not lead to disease because they may be recessive, or sometimes the mother has a rare genetic variation that is not enough to cause disease. The father has another one, but then when they combine in the child, they are enough to cause disease. But at this level is only a suspicion. These common variants, you wouldn't be able to pick up individually because there are hundreds of them, and people are trying to figure out, like, to develop something called a polygenic risk score, in which you take all of these individual variations and you add them up, and it gives you a score that could predict, whether the risk of autism, but we're not there yet. That's something that we're still working on. Many of these mutations are de novo, so the father or the mother doesn't have them, but they occur in the sperm. And when they occur in the sperm, they of course are sort of transmitted to the child. So these you wouldn't pick up by genetic testing of the parents, but when, let's say, the child has autism, is diagnosed with autism, you perform genetic testing on the parents and you perform genetic testing on the child, and you see that the child has a specific mutation, but the parents don't have it, then you can assume, oh, this is a, um, a spontaneous de novo mutation. So, the risk of having another child with a de novo mutation is very small. But let's say a child has autism and we test the parents and the child and we see recessive mutations in the parents and the combined mutation in the child, then there is a risk of having another child with increased autism risk. There is the real risk. Or if, other autism, because as I discussed about the overlap between schizophrenia, autism, ADHD, and these genetic risk, what it also means is that if in the family there have been several people with other conditions, not necessarily autism, but like schizophrenia or bipolar disease, then there might be an increased risk of having children with autism. So in that case, genetic testing could also be helpful.
Well, thank you so much, Dr. Penzies, for sharing how advances in genetics, brain imaging, and drug development are offering new hope to individuals with autism and related neurodevelopmental disorders. For all the attendees, thank you so much for joining. An email will go out next week to registered attendees with a link to recording of today's webinar. We hope you have a wonderful day, and thank you so much for joining us.
Well, thank you, everybody. Thank you for your attention. And if any of you are interested in more or learning more about the Autism Center and the research we are doing, please feel free to follow up with Eric or me, and we can take it from there.
Thank you so much, Dr. Penzies.